Okay, thank you so much for inviting me. I really am so happy to be back here in Barcelona. The last time we were here was 2019, and it's wonderful to see you all in person. Today, I'm going to talk about something new that I am not as confident about because I've had to learn about this whole topic. So we're all students in this. Okay, so we are looking for biomarkers for GRIN disorder. And first, I need to say what is a biomarker and why we're looking for them. A biomarker is an indicator of the disease status. So it can be an EEG, and it could be a blood test. For example, if you're being monitored for type 2 diabetes, they will look for your, they'll do tests for your kind of long-term exposure to high blood sugar with a simple blood test. And that would be a great biomarker. If we had something like this that was, we could do a blood test and we could measure where a patient is in terms of their severity of their symptoms and does it improve if we give them a beneficial treatment. So ideally it would be something that's not very invasive, that would be inexpensive, and that gives us a quantifiable number in a different way than the rating scales do. So right now we're mostly relying on the rating scales, which of course are going to always be very valuable, but it's nice to have a more objective measure to go alongside with that. And Dr. Garcia mentioned that that she would also like that. So I think that the best clinical trial design would include both. Interestingly, a biomarker doesn't necessarily have to be connected to the most disabling aspects of the disorder. So, for example, we could look at something in the blood that is a difference that doesn't necessarily cause a problem to the patient, but still is kind of a byproduct of the real problems that are going on. We can also use biomarkers to help with diagnosis. So for example, there might be different changes that happen if an NMDA receptor is overactive or underactive or has a complex phenotype that's somewhere in between those two. And the nice thing about these blood measures or EEGs is that they're less vulnerable to the placebo effect that that rating scales can have, because as Dr. Garcia mentioned, everyone wants to believe that we're doing something that would help the patient. So it's very tempting to see benefits, and it's nice to have a blood test that would go along and corroborate that impression. So we're working with a few different mouse models. They are all variants in the GRIN1 gene. We are interested in 2A and 2B, but we have not worked with those yet. What we have is a knockdown model that is not really representative of any patient. It's a dramatic decrease in the amount of GRIN1, but it could be useful for studying what happens in haploinsufficiency, because the receptors that are made are, biophysically normal. There's just fewer of them. But more recently, we've been studying these two patient variants. One is Q536R, both of them in the heterozygous state, just like the patient. We think that this one is a loss of function because it's a missense variant that affects the ligand binding domain, and studies in cells indicate that this causes a 3,000-fold reduction in the potency of glycine. So really, they're not responding at all to glycine. The other is a more complex variant. It's Y647S, and we originally thought this was a gain of function variant. But then with further studies, we see that in some contexts, it's a gain of function. In other contexts, it's a loss of function. So it's really very complicated. And this is a variant in the third transmembrane domain that's right in that channel 4 region. So one of the ways that we could go about looking at biomarkers is to, to collect serum from the animals and apply that serum to ELISA panels. And actually, Christian Branger suggested we try this company called Olink, which is based in, I think they have a couple of branches, but the one we went to was in Boston. So we sent samples to Olink, and they conducted an ELISA panel of many known biomarkers for other diseases. And we did see some that were significantly different. So for example, in the Q536R, we saw reductions in troponin and interleukin 10. In the Y647S, we saw increases in lipoprotein lipase and calcium binding protein S100A4. What I want you to see, though, is that there's a lot of overlap between the wild type and the patient variant. So I'm not convinced that these will be good biomarkers because they're not that different from wild type, and we'd like something that really stands out as being very different. So we continue to look in other ways. So I'm not going to say much more about the serum. I guess one thing that I should say, though, is why are we doing all of this in mice? And when we started this, we actually didn't have repositories of the patient serum samples. And now we do, thanks to Simon's. And thanks to other Combined Brain is helping to collect these samples. So I think that really what we need to be doing with these serum samples is we need to be testing these in patients. And I just want to say thank you to all of you who have donated to that. And I think we also need to have healthy controls. So if you have children or if you are able to donate, those are also valuable, just so that we can compare the patients to genetically related people who don't have a GRIN variant. Okay, so the other thing that we've been doing is to study the blood cells of these mice. And we did this because there are low levels of NMDA receptors on different blood cells, which I think is not really fully appreciated. But there are actually many papers where they have detected the mRNAs for these different subunits, and also they've detected functional NMDA receptors using drugs that activate and inhibit those receptors. So we know that they're there, but they're in very low levels, and they're not in all of the blood cells. So for example, they might be in a fraction of the red blood cells or the megakaryocytes or the B cells or T cells, and I'll show you some of that data. So I think that that's why they're not studied that much in the blood cells. So my collaborator, Maggie Caleb, published a paper in Blood where she showed that a cell-selective knockout of GRIN1 in megakaryocytes led to deficiencies in platelets and problems with clotting. So that was seen in mice. And that was very exciting because we thought, oh, well, that sounds like a really good biomarker. So this was really the reason why we started, actually, was her publication. And so we started with the CBCs, and which is something that probably every child has had, and all of us have probably had at one time or another when we've gone into the hospital, because these CBCs can tell a lot for physicians about things that might be going on. So what we saw, for example, in the Y647S is that they have increased numbers of red blood cells. Now, they are not dramatically increased, so you can see that there's still a lot of overlap. But if you test enough samples, you can see that these are significant, and they could be telling us something about the biology of what's going on. So these CBCs in and of themselves are probably not good biomarkers, but we can follow up and try and understand what's going on, and maybe it will reveal a factor in the blood that is a better biomarker than the ones that we saw with the ELISA. So that's our goal. So what we see is, I'm not sure. Okay, great. So we see increased red blood cell numbers, increased hemoglobin and hematocrit, and increased platelet. So this might indicate that in the blood cells, the Y647S is acting in a gain of function manner. Because the reason why I say that is that the knockout caused kind of an opposite phenotype with fewer platelets, for example, but the red blood cell phenotype was actually pretty surprising. So then my student who discovered all of this, Sylvia Okafor is her name, she went on to look at erythropoietin because this is a hormone that's needed to make red blood cells. And since they had more red blood cells, we thought that there would be more of this EPO-A in the serum, but in fact, there was less. And when she went further and looked at, the phenotype in males and females and looked at this data in both, this phenotype was only seen in the male mice, and in the female mice, there was no difference. So we still don't have an explanation for this. But then when she went back and looked at the CBC data, dividing all of that information out by sex, it turned out that the changes that we were seeing were really driven by changes in the males. So, this phenotype seems to be specific to the males. Now, when you put in this information, and again, this was all Sylvia's detective work of seeing increased red blood cells without changes in erythropoietin, that is a hallmark of a condition called polycythemia vera. So polycythemia means more red blood cells, and this can be done, for example, with when you have low oxygen levels. You go to the mountains and, people train in the mountains so they can get more EPO and more red blood cells. So this is kind of normal. And then this polycythemia vera is increased blood cells in the absence of an increase in EPO, and it's not due to hypoxia. So this is really what the picture that we have in those mice, which is quite surprising, and we still need to do more studies to understand what that is. But we're hoping that in uncovering that, we can get at some of the biomarkers that we're looking for clinical trial design. Okay, so then on the other side, we also studied the Q536R mice. And here we saw changes in red blood cells in their size and in their hemoglobin concentration. Because of this, Sylvia went and looked at the places where iron is stored and the functional pools of iron in the red blood cells and in the bone marrow. And this CBC panel and the studies that she made indicates iron deficient anemia. I'm going to show you this data. She did look at things like oxygen capacity and clotting time, and those were actually normal. So unlike the cell-selective knockout, there were not clotting deficiencies in this loss of function variant. So it's not as extreme of a problem as in the complete null variant, null mutation, actually. Okay, she also looked at blood viscosity and saw that it is increased. So the red blood cells are increased in size, and they don't have the same deformability, which is their ability to kind of shrink as they wiggle through little capillaries. I wasn't sure if I would have time to show all of this data, so I kind of have the summary here. Now I'm going to walk you through some of the CBC data that we have. So this is showing this increase in the volume of the red blood cells, that they're bigger, and there's a wider distribution in their width. So there are some that are smaller, some that are bigger. And also this reduction in the concentration of hemoglobin within those cells. So they're bigger, and it could be that there's the same amount of hemoglobin and it's just less concentrated. And there's also a trend for fewer red blood cells. So it does kind of seem like an opposite phenotype to what we saw with the Y647S. So this is the histology that I mentioned. There's a stain that you can do for stored iron, which is iron that's complex to ferritin. And I'm not sure if you can see this, but okay, it's these little blue speckles that you see here in the spleen. And what you can see is that in this spleen of the wild type, you can detect a certain amount, and then in the Q536R variant, there's less. And so this was quantified using, circling the area and then the intensity. So this is done with QPath and a software and a blinded fashion. So Sylvia didn't know which genotype she was working with when she did the scoring. And you could see that there was less stored iron in the spleen, which is kind of a storage tissue for iron, and also in the functional pools of iron, which is in the bone marrow. She studied the sternum bone marrow and stained that for hemosiderin as well. So this just kind of puts together the evidence that she has right now, that the Q536R have a picture that looks like iron deficiency anemia. So the storage pools for iron are tissues like the spleen, the liver, and lymph nodes, where the functional pool of ferritin and iron, stored iron, is in red blood cells, in bone marrow, and in cardiac and skeletal muscle. So we haven't looked at all of these, but what we see is that in the first stages of iron depletion, you would only see the change in the storage pool, but there would be normal levels in the functional pool. In iron deficiency, there's reductions in both the storage and functional pool. And then in iron deficiency anemia, that's where you actually have some red blood cell loss. And so this would be considered a very mild case of iron deficiency anemia, because really the red blood cell count doesn't reach the threshold, but there is a reduction in hemoglobin. So it's not really clear to what extent this might contribute to any symptoms. Again, as a biomarker, it doesn't necessarily need to contribute to the symptoms to be valuable. But we all know that, supplying oxygen to the brain is very important. And if there is anemia, that would mean that there's perhaps less oxygen that's being distributed to the brain. Okay. The other thing that we've done working with immunologists is to start to do flow cytometry to look at the white blood cell populations. So everything that I talked about before was about the red blood cells and platelets. But when you do the CBCs, you also look at your white blood cells. And when we did this, we did see some changes that we then followed up with a more sophisticated spectral flow cytometry. And what I would like to show you is this was only done so far in the Q536Rs. But down below, at the same time, we labeled for NMDA receptor using an antibody against the GLUN1 subunit of NMDA receptors. So what this is showing is that in these cells, the percentage of the naive CD4 positive T cells that actually express NMDA receptors or express the GLUN1 subunit on the surface. So you can see that it is a small percentage for most of these, less than 10%, 1% often, but in this class of T cells, it's actually 40%. So that was surprisingly high. But in those cells, we actually didn't see a difference in the number of those cells, but there could be a difference in the function of those cells. We did see differences in the amount of naive CD4 positive T cells and naive CD8 positive T cells. So it may be that there's less of of this transition from a naive T cell to one that is responding to an immune challenge. So we will see whether we can detect any differences in their immune system by giving some challenges to the immune system. These mice are stored in a very clean facility where they don't see a lot of pathogens. Okay. So what have we learned so far? Just to summarize what I talked about, the CBC assay is a potential source of biomarkers, and I am very excited to see whether these things hold up in patients. So, I definitely want to take advantage of what Simon Searchlight has done and apply to get some of this patient de-identified data and see whether some of these findings hold true, because I'm hoping that CBCs might be included as as a very common medical test that probably, patients have had to do at one point or another. Also, we see that the Y647S variant has increased red blood cells without increased erythropoietin, and that's consistent with polycythemia vera, but it's only observed in the males. The Q536R variant has decreased red blood cell hemoglobin, decreased iron stores in both the spleen and bone marrow that would be consistent with iron deficiency and potentially iron deficient anemia. And also, I've shown you that NMDA receptors are present in white blood cells, and that the Q536R mice have increases in naive CD4 and CD8 positive T cells. So what are we going to do next? Well, we would really like to see whether similar trends are observed in the patients. We also want to see whether any of these blood changes would be good indicators of the status. So for example, we are testing different therapies on these mouse models, and we want to see, if the mice seem to get better in terms of their electrophysiological profile and their behavioral profile. Do these blood measures change in a way that correlates with these more important measures? We would like to understand this mechanism for the reduced iron stores, and also understand whether this iron dyshomeostasis might contribute to any of the symptoms of GRIN disorder. And understanding why we're seeing this effect only in males. It's interesting because Dr. Viklicki showed that some of the phenotypes of the GRIN2B were seen only in the males, or so understanding why this is I think very interesting. And finally, we're going to be studying the immune system further with challenges to the innate and adaptive immunity using things that are like inflammatory stimulators, and things that would stimulate the production of antibodies, like a DTaP vaccine that that children receive. So I want to thank the people in my lab who've contributed to this work, especially Sylvia, who is the detective and a fantastic graduate student in the lab. In addition, I have some wonderful collaborators that I want to thank. The Collingridge Lab has helped a lot with the electrophysiology that we're learning about these mice using this. We're doing some prime editing studies with Basil Hubbard's lab. Evelyn Lamb is also doing some electrophysiology studies. These mice were actually made at NHGRI, I think, National Human Genome, sorry, there's a typo there. And I want to thank my collaborators at Toronto, especially Landon Edgar and Thierry Malave, who did the immunology part of this, and my collaborator, Maggie Calev, who is a clinical hematologist and is kind of looking over all of this and making sure we're not doing the wrong thing. And I see I've misspelled Auckland, but she's in New Zealand. Okay, and that's where I will stop. Thank you. Okay.